Now that we've gotten through the carbon cycle and talked about oxygen dynamics, and redox, we can talk about the other elemental cycles. And these two images are related to that. The one on the left is from Mammoth Hot Springs, and these massive uh, white streamer things are actually sulfur oxidizing bacteria. And the first step of taking sulfide and combining with oxygen and then results in elemental sulfur. And that's the biggest energy gain in that whole step of the process, although eventually it'll go all the way to sulfate. And that then gets deposited inside the cells of these organisms and leads to this white deposit that you can see on this. And then also there's carbonate depositing as well. You can kind of see the terraces because it's super saturated and when it comes out from the hot springs. So if you ever get a chance to go to Yellowstone, definitely take it, because you'll know a lot more about it when you get through this class. We'll talk about hot springs and extreme environments. It's a great place to think about sulfur cycling and nitrogen and carbon cycling and all those kinds of things, as well as having systems that are ruled by microbes. And on the right-hand side is a electron micrograph of the cyanobacterium, and it has a heterocyst, which is a site of nitrogen fixation. We'll talk about that in a good bit of detail. In a vegetative cell, which is just does photosynthesis, oxygenic photosynthesis. And you can also see a gel that's surrounding it, and a little bit of bacteria that's growing inside of that gel. So as I said, we'll talk about nitrogen, sulfur, phosphorus, silicon, iron, other trace elements a little bit, and then go back to that gradients of redox idea, nutrient cycles, and how they all interact with each other. So this is chapter 14 in your book. We'll start with the nitrogen cycle, one of my favorite topics of all time. The first thing always is we talk about the forms of nitrogen, and the inorganic nitrogen forms are listed across the top. Nitrogen gas makes up most of the atmosphere, and there's a little bit of nitrous oxide in the atmosphere, and we've talked about that just a little bit because it's a greenhouse gas, but it also is involved in destruction of ozone in the upper atmosphere. Ammonium is NH4. Ammonium can either be in an ionic form when you are in acidic conditions. If you take ammonium and make it basic, though, it goes to NH3, which is ammonia gas. That's a toxic gas. That's why you do not want to mix Clorox and ammonia cleaner, because both the chlorine gas and the ammonia gas will come off, and it's toxic and it's dangerous. So you don't want to do that. You're converting that ammonium to ammonia gas. That's also why when you go by a feedlot, and it has, part of the reason it has a real sharp smell to it, because the cattle will excrete, and the urea will be converted to ammonia by bacteria. And ammonia, if you soil somewhat basic, the ammonia will come off, and you can just smell that. That's part of the reason that we have increases in atmospheric loading of nitrogen in this part of the country, because of feedlots emitting ammonia into the atmosphere. Then it gets into the rain, and the rain is acidic, so it can turn it back into ammonium ion. Nitrite, we'll talk about that in more detail. It's part of the nitrogen cycle, and it's a quite toxic little molecule. And nitrate, which is a very common form of nitrogen when there's high levels of nitrogen loading and oxygen. Then there's all the organic nitrogens that include the amino acids, it includes the nucleic acids, and a number of other compounds like urea that I just mentioned as well. And we're going to talk about the major nitrogen fluxes. There's uptake first, mineralization. Denitrification is an important one for blowing off nitrogen to nitrogen gas, nitrification, dissimilatory nitrate reduction, and nitrogen fixation. So we'll start with the uptake here. And we see on the top, we have nitrogen gas and ammonium and nitrite and nitrate. And ammonium is the form that nitrogen has to go in to get into building proteins. So this is all this stuff down here. You don't have to worry about this very much, that this GO-GAT pathway other than the main fact that ammonium is what's coming in here and to build the base amino acids, which then go on to produce proteins. But then given that we have to take ammonium into cells, if, well, it's an organism that can use ammonium like a bacterium or a plant, that then sets up the situation where if they're going to use some of these other compounds, they're going to have to convert them to ammonium before they can be assimilated into the cells. And we see that nitrogen gas takes 6 ATP plus some free electrons, and it takes energy to produce electrons. Using nitrate, you have to convert it. It takes 8 electrons, which also takes some energy. So this is the first question of the day. Why does this diagram suggest that nitrogen-fixing bacteria should downregulate genes for nitrogen fixation if nitrate or ammonium are available? So we've got some good answers here. Yeah, the 6 ATP up here. It turns out that nitrogen fixation is perhaps the most expensive, energetically expensive chemical reaction that organisms do. It requires 6 ATPs, which is a lot. And to get to ammonium, they have to convert to ammonium, and then they'll take it in. This turns out to be a lot more than the energy it takes to use nitrate and to ammonium. Still, if ammonium's available, most microbes will use ammonium first, even if there's not very much of it, because they have to convert the nitrate to ammonium before they'll use it. So the first choice of many organisms is ammonium because it has the lowest total energy cost. Second choice is nitrate. Most nitrate doesn't hang around the environment very long. It's rarely very high, so they'll have to convert it through that way. And then the last choice is a specialized organism, such as the bacteria and the cyanobacteria, which are able to fix nitrogen. So as I just said, nitrogen gas to ammonium is very expensive, and only bacteria are known to do this. They keep saying that they're going to put the genes for nitrogen fixation into plants, and they don't seem to be able to. What are the plants that take advantage of nitrogen fixing bacteria? Anybody remember that? They're not real important in freshwater ecology. There might be a few, but legumes, good, Emma. Our horticulturalist has the plant answers always. Right, so the legumes you have nodules, and they put bacteria in them. Nitrogenase is the enzyme that allows for nitrogen fixation, the conversion of nitrogen gas to ammonium, very sensitive to oxygen, and it'll just basically knock it out. And so there's a whole bunch of adaptations to protect it, and we'll talk about these, and then starting the next slide. In addition to this, in the nitrogen cycle, lightning also fixes N2 to nitrate in the atmosphere. It takes a huge amount of energy. So nitrogen gas is less oxidized than nitrate. So it does have potential energy, but it just takes so much energy to convert it that most organisms just can't or won't do it. And this is, the lightning, it has a lot of energy, right? So it can actually zap the nitrogen hard enough to N2 gas to turn it into nitrate. Nitrogen fixing cyanobacteria can be really important in light nitrogen cycles, particularly when you load a lot of phosphorus in. It allows them to catch up to that and really make some impressive blooms. So here's cyanobacterial heterocysts, and I should change this slide because they've renamed heterocysts heterocytes, and that's just something that's happened over the last couple of years, and I have trouble changing what I learned as a undergraduate and a graduate student. So we can call this a heterocyte or a heterocyst if we want to. And vegetative cells are producing oxygen in the light all the time. What from? Why are they producing oxygen in the light all the time?
Can you ask it again?
So vegetative cells are producing oxygen in the light. So we have a cyanobacterium, it's producing oxygen in the light all the time. What's that process that is creating oxygen in as a result?
Oh, photosynthesis.
Yes, good, photosynthesis, right? So photosynthesis actually has two um, units, photosystem 1 and photosystem 2. And photosystem 2 is where the oxygen is produced when they split water. Photosystem 1 just generates ATP from the electron energy. So the first thing that heterocysts or heterocytes do relative to your normal vegetative cell is they turn off photosystem 2. So they're only generating ADP and ATP. And they're collecting a lot less light. So when you go and look at the algal cells, you'll see that the vegetative cells look nice and green under the microscope, and the heterocysts or heterocytes look yellowish. And that's because they dumped a lot of that photosynthesis off. Because they don't want to create oxygen inside of this heterocyst because it'll deactivate the nitrogenase. In addition, they have a series of gel layers around the outside that retards inward diffusion of gases, so the oxygen can't diffuse in very well. And they really crank up respiration. And so the oxygenic photosynthesis in the vegetative cells, they transport organic carbon into the heterocyst and respire at really high rates. They drive the oxygen down in concentrations down inside of this heterocyst. Then the other thing that this does is it takes the ammonium and immediately reacts it with glutamine into glutamine, and then this goes through that GOGAT process that we had on the prior slide. Then they transport that out continuously into the other cells. So the vegetative cells are pushing carbon in, and in return, the heterocyte is pushing nitrogen out into the vegetative cells, so it allows them to grow. In addition to these adaptations, you saw the picture at the very beginning where there were bacteria embedded in this gel. It turns out those bacteria are also respiring, and they actually use up oxygen, and the cyanobacterium will leak out organic carbon and stimulate those bacteria to respire outside. So they'll have an additional protection of their nitrogenase. There's some other tricks that cyanobacteria can use. They can make big clumps where the center is not photosynthetic, but the outside is, so they don't need a heterocyst. They can fix nitrogen in there. And there's some examples that we'll talk about when we talk about symbiotic bacteria that are helping fix nitrogen for other organisms, and there's some adaptations for that as well. So some other nitrogen cycling, we talked about nitrogen fixation. Nitrification is the oxidation of ammonium to nitrite and then nitrite to nitrate. And this only happens in environments that have oxygen in them. It is a chemoautotrophic process. That is, the organisms use the ammonium oxidation and the nitrite oxidation to generate ATP, and then they use that ATP to fix carbon dioxide to organic carbon. So it's a chemoautotrophic process. This has to happen when there's oxygen around, because ammonium and nitrite only have potential energy relative to nitrate in high oxygen environments. The semi-opposite of that is denitrification, or maybe you can think of it as the opposite of nitrogen fixation, is using nitrate as an electron acceptor to oxidize organic carbon. So this is one of the first respiration, if you will, using an inorganic carbon other than oxygen as you go down the list of things that you can use to oxidize organic carbon. It's the most energy efficient after oxygen. It yields nitrous oxide and nitrogen gas, and actually nitrification leaks a little nitrous oxide too. The nitrous oxide is really important because it's a greenhouse gas and it has global importance in that. But eventually they want to get to a nitrogen gas that yields the most energy. Under extremely low redox, the ammonium has lower redox than nitrogen gas, and so you can get the conversion directly to ammonium from nitrate instead of N2. And remineralization, and sometimes in anoxic it's called ammonification, is just taking organic N and turning it back into ammonium. So here we'll put the nitrogen fixation and all the other fluxes on the scheme with oxygen on top and anoxic on the bottom. And we have organic N on the furthest left. Ammonium being more reduced than nitrite, which is more reduced than nitrate. Nitrate is the most oxidized form. And we see on this oxidation state that nitrous oxide, nitrogen gas, so it actually goes ammonium is the most reduced, then nitrogen gas, then nitrous oxide, then nitrate, and then nitrate. We talked about the assimilations, right? So you can assimilate nitrate, turn it into nitrite, and then to ammonium, and then take it into the cells. So assimilation can occur in an oxic or anoxic environment. So that's why the arrows for assimilation are drawn along that boundary, just because they can happen in both places. We talked about nitrogen fixation, taking into gas, and really it bounces through ammonium, but it ends up as organic N. And this happens in anoxic conditions. So what is it that causes nitrogen gas to have anoxic conditions? Just to see if you're remembering a few minutes back in my lecture. Anybody remember what the enzyme is that allows nitrogen fixation? It's nitrogenase, right? And what is it? Yeah, nitrogenase is sensitive to oxygen. So that's why I threw this down here. So even organisms that are in oxic environments, such as legumes and the nodules of the nitrogen fixing bacteria in the nodules of legumes, or heterocytes, they're in oxygen containing environments, but they're both protected against oxygen. The legumes, if you cut them open, they have this hemoglobin that gloms into the oxygen and keeps the oxygen off of the rhizobia, which are the nitrogen fixing bacteria in that. Yeah, so I put that down here. And then we have nitrification up here, right? Ammonium going to nitrite, and then nitrite going to nitrate. Is that going with or against potential energy? With, good. And then we have denitrification, and if it gets really low redox, we get dissimilatory reduction of nitrate to ammonium, DRNA, and this is the more, it gets favored under really low redox. There is one flux in here that I've not talked about yet, and we won't necessarily cover it, but oxidation of ammonium and nitrate or nitrite to nitrogen gas is another flux out, and it's just a way to generate more energy, and fairly recently described, but we won't spend a lot of time on that. Okay, so this is the nitrogen cycle. One of the other things that can help you think about these and understand them is I left one off, mineralization, right? And mineralization can happen in oxic or anoxic environments. Most aquatic organisms will excrete nitrogen as ammonium, even in anaerobic conditions. However, many terrestrial organisms don't. They convert their waste into urea. The reason for this is that ammonium is a toxic ion. It's toxic to organisms, and so if you build up a lot of ammonium, you are going to poison yourself. This is why sewage treatment plants are trying not to release ammonium into rivers. They're regulated, and they're regulated based on the amount of ammonium in the pH, because it's actually most toxic as ammonia gas. So if it's more acidic water they're dumping into, they can get away with dumping more ammonium than if it's more basic water. Aquatic organisms can just dilute out the ammonium. Fish exchange across their gills not only oxygen and CO2, but they also can drop ammonium out into the environment across their gills. In contrast, we don't have unlimited amounts of water. So if we build up our urine inside of our body, right, if it was just in the form of ammonia, we would be poisoning ourselves. So urea is a lot more neutral. So that's why we push it into urea, concentrate it, so we don't have to waste a ton of water. And many terrestrial organisms use that kind of trick. In contrast, mineralization of organic in anoxic conditions can be sometimes called ammonification. This is part of the reason that and this is oftentimes comes about breaking down proteins or amino acids. It does not smell good to us. Ammonia does not smell good to us, ammonia gas. And it makes, for most people, it makes food taste rather distasteful to have a high concentration of ammonia in it. There is a Chinese dish that is sort of a fermented dish that's high in ammonia, and I don't like it very much, but it tastes kind of like a feedlot, I think. So some people do like it. Some people like sulfide smell similarly, but for the most part, it's something that's distasteful. And of course, it's organic and recycling. We take amino acids and nucleic acids and break down proteins and DNA and RNA in our guts, and then take them across the gut wall and then build them back up, as do fishes and invertebrates, and most animals are unable to do this straight assimilation into the organic and from ammonium. One of the really big issues related to nitrogen is the pollution of nitrogen by nitrate. And nitrate's not allowed in drinking water in the United States. Over 10 milligrams of N in the form of nitrogen, N in the form of nitrate per liter. And the reason for this is it can lead to methemoglobinemia. And I sometimes ask students to say methemoglobinemia 10 times fast, but basically, anemic, right, means low oxygen, right? And it's because the hemoglobin gets bonded by nitrites. Yes, and right. So Emma says, "Whoa, there's nitrates in meat, deli meat, right?" Yes, this is part of the problem. This is, and we'll get to that. That's a really good observation. This is why, like, cured meats are lead to colon cancer in part. And the problem is that it can be converted. They can be converted to carcinogenic nitrosamines in the stomach. So that last point is important. Methemoglobinemia, blue baby syndrome. What happens is the nitrate is converted to nitrite, and the nitrite then bonds to hemoglobin. If an infant is fed formula that's high in nitrate and not nursed, right, then the nitrate can be high enough that they can really get all their oxygen bound up, and so they look blue. Their hemoglobin is not getting oxygen in it and getting that nice pink look to them. Or reddish coloration to their blood underneath their skin. Okay, so here's a nitrogen cycling question. One of the places that it's really common is below feedlots. So in Kansas, one of my colleagues went around and sampled a bunch of wells and in Kansas, and found that of people that are just on well water in rural areas, a very high percentage of those wells have nitrate that exceeds the standards that are allowed for drinking water. But when he talked to them, they're like, well, we don't know why this happened. And then eventually they'll say, oh yeah, like 20 years ago, the guys before us had a little bit of a feedlot here, or a winter pen where they held their cattle that was right over where our well is. However, most of the excreta has organic N or ammonium as a dominant form of nitrogen, basically urea, which then gets broken down into ammonia. So why is it that nitrate ends up being so high in what is it that allows nitrate to be so high? So let's go back up as we ask that question and look at the nitrogen cycle. And Emma said the bacteria change it. Yeah, right, exactly. So organic N, right, here is excreted as ammonium or maybe as urea, which is then mineralized to ammonium by the microbes, right? Okay, so then what process must be leading to formation of nitrate? What's the only way to get to nitrate from here? Good, Trevor, nitrification, right? So there must be some part of the groundwater that is in contact with oxygen, right? Because if it was anoxic, it would be going against potential energy. So you do have to have a situation where you have at least the water flowing into some part of the groundwater that has enough oxygen that the nitrification becomes dominant. And so then you can get high nitrate concentrations in drinking water and surface water, streams, and such in agricultural areas. So this is why understanding the nitrogen cycle is important. And if we look at nitrate intake in different places in the world, USA is pretty low for the most part. And this is just happens to be in millimoles of, well, we've talked about what a mole is per person per day. And USA and Sweden are pretty low, Norway's pretty low. But we get into some of these countries that have really intensive agriculture and lots of deposition of nitrogen onto cropland, and you get a lot of nitrate consumption, and that the gastric cancer rates are significantly higher. So this goes back to what Emma was saying. It's also related to the idea of eating a whole lot of cured meats, and that's why people don't recommend eating a lot of, you know, sausages and bacon and such all the time, because it also can increase rates of gastric cancer. So this is why the nitrogen is limited, and it's illegal for a drinking water treatment facility to put out high nitrate water into a domestic water system for that reason. It's also interesting, though, that because Europe has so much more intensive agriculture, their nitrate levels that are allowable in drinking water are higher than they are in the United States, because we don't have quite the intensity of agriculture stacked in with a lot of people as they do there. And Japan really hasn't. We can talk about how this plays out in a lake. And we have, this is a really eutrophic lake in this case. And this is a lake that has ice cover from January to March. And it becomes a little bit. So we've got, in A, we have nitrate, and in B, we have ammonium. And the blue colors are low concentrations, and the red colors are high concentrations. So with respect to the nitrate, right, we're having high concentrations in the spring when the lake is mixing. So what process, let's say ammonium comes in there, what's happening to the ammonium in the low concentrations? Ammonium concentrations become pretty low at that same time. What process would be taking this ammonium that's in the water in the winter time under the ice, right, and converting it to nitrate? Nitrification, good, Trevor. Aaron, good. Nitrification. So the nitrification is driving. There's a lot of nitrogen in the system. And then why is there so much nitrogen around the system in the spring in this area? What do you think is leading to high nitrogen in the spring? And a hint, this is an agricultural area. High total nitrogen planting, getting closer, Abigail. Fertilizing, yeah, fertilizer, right? So people fertilize their fields in the spring. And it might be building up in the soil throughout the winter. And we're talking about Michigan, right? So what else happens in the spring? Melting, right? Snow melt. So you get a lot of flushing through. And the extreme example of this was what they called weather whiplash that happened in the upper Midwest a few years back following the severe drought. So the spring following the severe drought, they went ahead and planted their crops throughout the year, and the nitrogen built way up. In the wintertime, it got converted over, right? Broken organic nitrogen, ammonium all got converted over to nitrate, but it just sat there. It rained or snow melted in the spring and causes flush of nitrogen and caused a huge cost to treating water downstream in the municipalities downstream there. And so it's led to legal battles or whether the farmers were responsible for that nitrogen input or not. And that's not completely resolved. Okay, as we move into this summertime, the nitrate levels are starting to drop off, right? And in particular, the ammonium levels are coming up. So what process would be causing the nitrate to disappear? Oh, good, Trevor. Denitrification, exactly, right? So the nitrate, the bottom of the lake, this is probably stratifying a little bit, right? And the bottom of the lake is getting anoxic, and so then the nitrogen can start driving off. And in addition, Emma wrote respiration, it's kind of close. The phytoplankton are probably growing pretty good in the summer, right? So they're taking nitrogen up at the same time. So it's going into the cells. So we have the stratification, and then late on the anoxia, it goes really low. The redox goes really low, and we get this conversion of nitrate straight to ammonium, DNR A in here. And then what happens here? Why is it all the same from top to bottom in November? What happens to the lake? Why is it, what's going on here that's different from here? I mean, we're talking about 1 month. The ice forms in January. So the ice hasn't formed yet. Ice forms here. Mixing, good, fall mixing, right? So it cools down by the end of September, beginning of October, and starts to mix top to bottom. And once it does that, then we get the concentrations are equal throughout. Then once it gets cold enough that the ice does start forming on here, it stops mixing and we start getting anoxic and moving over into this portion. So we talked about, we had a question about fish kills in the quiz this last week. So we go back, if you didn't get that one right, go back and review that idea.
We talked about nitrogen fixation being oxygen sensitive. Why does it being anoxic accelerate nitrification? Like, are those enzymes also sensitive to O2, or is it just like redox?
Why being anoxic doesn't, if it's really low redox, right, the nitrate, the ammonium has higher potential energy. The difference between nitrate and ammonium becomes so high that nitrate is used to oxidize organic carbon and it's converted to ammonium. Right, so from nitrate to ammonium. So first denitrification is going to be happening, but eventually you just get to the point where everybody's dumping everything into ammonium because that's the lowest redox compound. That's a good question. And then here's an example of some nitrogen distribution in a stream where we have this groundwater that's pretty constant, but the stream downstream has high nitrate in the summer, and then in the spring it has low nitrate. And this is an example of a stream that's next to cropland on Kanza, but the upland that's feeding it is all native prairie. So in the spring, when it rains, it's coming off of pristine prairie and diluting the nitrogen out. So here's the discharge, right? It's diluting that out. In the summertime, when the flow is low, the and the cropland's being fertilized, the cropland's been fertilized in the spring, the nitrate's moved its way down in through the groundwater, and it's coming out into the stream, but it's not being diluted out from the low nitrogen water from above. So we get this annual swing in nitrate in this particular habitat. Good news is that these croplands have been converted to tallgrass prairie in restoration experiments, and this nitrate value actually has fallen considerably in the last few decades since we did this. But we can see these big swings in nitrate in streams. I already talked about the weather whiplash thing. You go downstream of golf course, a lot of urban areas, people just dump a ton of fertilizer on their lawns because they figure out if I can put this much on, if I put twice as much on, my lawn will be twice as green. So they don't really control the amount of nitrogen that they put on, unlike a farmer who may add a little bit more nitrate than they need, but it does cost them, and they're talking about many acres of land, so they tend to be a little bit more conservative about how much they add. That gets us through nitrogen cycling and now into sulfur cycling. And sulfur is interesting because it's one of the most complex ones. And the reason for that is that there's so many different redox states that the sulfur compounds can take. So here's only some of the forms. Sulfide is hydrogen sulfide is a gas. It could also be an ion. Again, if you acidify hydrogen sulfide, the sulfur comes off as a gas, and hydrogen sulfide comes off as a gas, and it's not good. Elemental sulfur is yellowish or whitish, and you've seen that. Thiosulfate is a combination of two compounds of different redox of sulfur. Sulfate is the most common in oxidized environments. Organic sulfur, there's amino acids have sulfur as part of them. There's a few other organic compounds that have sulfur in them. And dimethyl sulfide is another one we'll talk about in a little bit. One of the things that happens if you take sulfide and put it into water is that it can be converted to sulfate without any microbial activity. So I mentioned that if you have a compound of ammonium and you put it in oxidized water, the ammonium will just stay there because the activation energy you get from ammonium to nitrate, even though nitrate has lower potential energy, and that would be the way the reaction would go, the activation energy is not so high for sulfur. In addition, just a second, we'll make sure what my time's at. Oh, my time's up, isn't it? Okay, so we'll pick up the sulfur cycle when we start again.